Our genome is not just a string of DNA code. It requires signposting to bring it to life, so that cells know which part of the genetic code to use and when. This is where epigenetics enters the picture. Epigenetics is a mechanism for controlling when our genes are switched on and switched off. Every cell in our body has an identical genome, but this information is interpreted in different ways to make over 200 different cell types, such as brain cells, heart cells, liver, kidney and skin cells. These differences in cell types are due to differences in epigenetic marks that cause the genetic code to be interpreted in subtly different ways. Epigenetics has been studied for many years. The term itself was introduced by Conrad Waddington in 1942, but it was mainly studied as part of developmental biology. More recently, the interest in epigenetics has grown enormously. This is because it may also help us to understand how our genome malfunctions to cause disease. For example, huge epigenetic changes occur in cancer, causing cells to lose control of their growth and proliferation. There are a multitude of different epigenetic marks on the genome, but the most commonly studied is DNA methylation. Some DNA methylation marks, including those which decide cell type, are laid down very early in development and are faithfully replicated throughout life. Other DNA methylation marks can be influenced by the environment around us, for example, by exposure to cigarette smoke, alcohol, stress, or other factors. These epigenetic changes to our genome may influence our risk of disease in later life. They may in fact explain how some diseases are a consequence of both our genetic makeup and our environment. Alternatively, these changes induced by our environment, our lifestyle or our behaviours may be harmless but act as an archive of experiences that we have encountered over our lives. Take the example of a mother smoking during pregnancy. Epigenetic changes can be detected in the blood cells of babies at birth who have been exposed to cigarette smoke during pregnancy, and some of these changes persist for decades. They are still detectable at age 50 and beyond. Epigenetics is an exciting new area of biomedical research. It promises to improve our understanding of disease mechanisms and so to help identify new treatments or ways of preventing disease. It can also help us to understand the importance of risk factors for disease and to predict disease more effectively.